Neural dedifferentiation of semantic knowledge representations in healthy aging. Neural dedifferentiation theory proposes that neural patterns of activation in response to stimuli are more similar with aging, and therefore cortical responses are less differentiable. Previous research suggests that neural representations of categories such as faces, houses, or objects become more similar in the visual ventral string in old adults. Similar findings were found in memory, motor system, and working memory, among others. Despite this promising research, very little attention has been paid to neural dedifferentiation in the context of language and semantic cognition. Though conceptual richness tends to remain stable or even improve during healthy aging, some behavioral evidence suggests knowledge domains may be represented differently with age. On the one hand, by using fMRI in combination with decoding and representational similarity analysis, our goal was to examine the aging effects on the brain representations of semantic categories. On the other hand, we wanted to know how this dedifferentiation is modulated by semantic depth of processing. In this study participated 27 young and 29 healthy older adults above 65 years old. Participants were presented to 18 living and non-living words and exposed to two alternating conditions. In the first condition, the read condition, they were instructed to attend and continuously repeat the word in their minds. In the second condition, the think condition, they were instructed to mentally simulate sensory motor properties associated with the word. In this study, we performed both ROI and whole brain searchlight analysis. Here, for the sake of time, we are just presenting searchlight analysis. First, we perform a searchlight decoding analysis across the whole brain by training a machine learning classifier to predict category levels of specific patterns of activation. We train our model with all examples of 34 of the 36 words and try to predict the label of all examples of remaining living and non-living words. But before continuing, a brief clarification for better understanding. Neural differentiation is conceptualized here as the performance of the classifier to distinguish neural representations associated to two or more stimuli. The more accuracy, the more differentiation. Conversely, the less accuracy, the more dedifferentiation. First of all, regarding within condition decoding analysis, word semantic categories could be significantly predicted in the think condition, but not in the read condition, meaning that explicit mental simulation provides access to more meaningful stimuli representations. In the think condition, significant decoding clusters were located in the ventral visual pathway, both in young and old groups. Also, it seems significant decoding extends across a larger amount of brain regions in the young group, as it can be seen in the figure. Despite those results, no significant decoding difference between age groups were found in any condition. Then, we wanted to address the specificity of category representations across different task contexts by means of cross-condition decoding analysis. With the same cross-validation procedure, we test how well a classifier trained in one condition categorizes never-seen concepts in the other condition. We only found cross-condition generalization in the old group. This generalization could be observed both from read to think and think to read reactions in the left middle temporal gyrus. When comparing difference in the searchlight generalization between age groups, we didn't find significant effects in any decoding direction. However, ROI-based ANOVA analysis showed a significant effect of age in the left middle temporal and inferior parietal lobes, as can be seen in red in the figure. These results suggest that category semantic representations are less differentiated between semantic processing levels in old adults than in young adults. Last, we explored the geometry of representational spaces using representational similarity analysis. We wanted to know where in the brain activity patterns resemble the most to the features geometry extracted from different compositional models. First, by using 3 pre-trained word embedding models, we extracted the 300-dimensional feature vector corresponding to each experimental word. Similarly, 3 fine-tuned computer vision models with a custom 300-dimensional layer included on top were used to extract abstract representations of 10 image references of our words. Then, for each word, the 10 image resulting vectors were averaged together. Next, we compute features dissimilarity between pairs of words using correlation distance as dissimilarity metric. Lastly, for each computer model, we perform a whole-brain search-like analysis on which we correlate these RDMs with the whole signal representational dissimilarity metrics at each brain region. As in decoding analysis, in both age groups significant correlations were found in thin condition covering regions of the left ventral visual pathway. Then, we compare whole-brain correlations between age groups in both experimental conditions. Interestingly, in thin condition, young showed larger correlations than old adults across all computer models in bilateral postcentral gyrus and surrounding regions. In that region, young adults seem to have a more similar representational geometry to that of computational models as compared to old adults. A potential interpretation to that result is that old adults have less differentiated categories in that regions when compared to last slide RDM. Together, our results provide some support for the differentiation models of aging in the context of semantic processing.